So this should be the second part of our special senses, and if you guys have gone in the order so forth, I would, after this, go forth and take the test. Um, don't want to wait until you can't really remember much of it. All right. So far in the special sense of things, we talked about taste, we talked about vision, we talked about all the other visceral senses, such as touch and heat and cold and all that fun stuff. So, continuing on through special senses, we're going to head on to hearing. All right. So, hearing is probably just as intense as vision is. Okay, and obviously it's a very key part of our survival, so of course there's much more to it than we think there is. All right, so as you know, hearing is what you're doing right now, is listening to my voice, is just the ability for you to perceive sound vibrations, all right, but convert them into electrical signals, which then shits on through to the nervous system. All right, and kind of like our scent, our taste, it can have these thresholds of either being really sensitive or not too sensitive. Ears, on the other hand, they are really sensitive. Okay, you can detect small vibrations. All right, so that's why if you're ever sitting in a room, you kind of start to hear that humming noise. That's coming from just a small, really sensitive vibration that you don't really, I guess, are aware on a large scale, but you are detecting it. Okay. And hearing, kind of with the other special senses, it also goes hand in hand with another sense, and we'll see that, and that is that it houses receptors for equilibrium. All right, so that's where it helps us get our balance from. We'll go over that when we get down the line. Now, the anatomy of the ear. Okay, the ear has three major regions. All right, the external, that's where your sound collection occurs. Okay, so the external, your middle ear. All right, so sound amplification occurs there. All right, so yeah, while you may be able to detect a lot of sound that's light vibrations, it needs to be amplified when it gets into our heads, okay, into our nervous systems. And then lastly, internal ear, which is where the sound transduction comes from. All right, so external sound comes in, middle, it's amplified, internal, it is turned into an electrical signal, transduced. So starting with your external ear, that's probably everything you can put your finger in right now when you're playing with your ear. All right, we got three main regions. One is our elastic cartilage that funnels sound waves. So that's the kind of elastic, crunchy cartilage you have on your ear on the outside. All right, they're called the auricula or the pinna. And then we have our ear canal, also known as the external acoustic meatus. Now the ear canal it acts as a canal. Instead of water, it's helping to direct sound via bouncing off the canal into the eardrum. All right, and the eardrum is actually a part of the external ear, so that is called the tympanic membrane. And the eardrum acts as a drum because a drum is mostly hitting and causing vibration, whereas the eardrum acts as that. Sound hits it and it vibrates. Okay, and what we'll see is that vibration is key for us to have our hearing. And so altogether the functions is exactly as I said. It collects and it channels sound waves, but it has some other roles. Obviously one of them is that eardrum. It gives you the separation of your external and middle ear. But a part of it that we didn't talk about, the three main structures, is a gland. Okay, and this gland is key for us because kind of similar to what we saw with vision and the eye and kind of keeping it clean and protected, our ears have the same function. All right, so we have a specific gland that produces earwax, and so this earwax acts as kind of like you can say like the first line of defense in our ears. All right, so it's there to help to trap any toxins, any viruses, any parasitic, anything that may come to harm our hearing or our body itself. Okay, so the earwax is actually there to assist us, to protect our ears. So, as said, external ear, right? Our little crunchy elastic cartilage on the outside, our canal, okay? And then our eardrum, all right? Because remember that separates inner ear, outer ear. And of course, our lovely depiction, right, of earwax. And now, earwax is not the only kind of protector here. Similar to smell, we have hairs in our ears, whether people like to admit it or not. But that is actually there again, similar to earwax, to be this protector beneficial. But it also comes into play when you have touch, or you'll see in a little bit equilibrium. All right. Now after that glitch, so we've gone from the external. Now we've gone into the middle. Okay, again, there's a variety of structures that play a key role. We have our tympanic cavity, which is just the air-filled space that's actually in temporal bone. All right, and auditorial ossicles. So we have three of them: malleus, incus, and stapes. All right, so these guys do exactly as you would think they would do. Okay, they are there to help with auditorial, you want to say, perception or intake, and we'll see in a little bit when we get to the physiology of hearing exactly how. Okay, and on the side of that, two of the things we have is our round window, or you probably heard of as a cochlear window. Okay, and that's there to actually help release pressure waves that were, your ear popping, right? It's there to help exist with that pressure being released and whatnot. And lastly, on the side of that of releasing pressure and when it needs to be, is we need to be able to equalize pressure all together. So that is when our auditorial tube within the middle ear comes into play. Now, external ear, it was mostly just this fibrous cartilage-based tissue. Now we actually have some muscles when we get into the middle of the ear. Okay, so we have a tensor tympania, or CN5, or stapes. Okay, so each does their own job. One tenses the eardrum, all right, which helps with, in a sense, you can think of like sharpening the sound to yourself. The other dampens or kind of stapes down the vibration, which can be key when it comes to adaptation. Okay, so together, these structures and these muscles work to either help amplify the sound vibrations, okay? Because remember, the whole point of the middle of the ear is that we want to intensify the sound vibration. So they all work together to amplify the sound vibrations about 20 times more than what they came in with, okay? But it also is there to help protect from too loud or too prolonged loud sounds. So if you're in a stadium that everybody's cheering, your ears are adapted to try and help protect your inner ear from the prolonged sounds by dampening the vibrations you're getting from all the crowds around you cheering. All right, so middle ear is not just there to amplify sound, it is there to protect as well from vibrations and loud sounds. Now, this is again just a depiction of what I'm talking about when I say middle ear. You have our gap, our cochlear gap, the cochlear window, which is there to basically help with kind of recognizing, I guess, the pressure changes, one way to say it, as well as our audio tube, as we had said, that is there to balance the pressure, equalize the pressure, whereas the cochlear is there to help to release the pressure. This is where you get your ear popping from, right? Pressure changes and the balance that's needing to be made. So, inner ear. Okay. Now, the internal ear is where the real magic gets done when you think of senses, right? We get our sense, we go forth and find our way to a central nervous system to tell us what it is. This is when the internal ear starts to do exactly what you probably think. And it has structures as well. All right, cochlea, which is actually there for the hearing, right? And then vestibular and semicircular canals, which we'll see they come into play when we talk about balance, which will be our final sense we talk about. We have fluids that are in it, two different ones, each varied by location. Okay, it's the main difference. Okay, whereas you have some that are in the cochlear duct, the endolymphs, and the perilymphs where are in two other locations. Now the spiral organ, sometimes referred to as the organ of Corti, that is on the basal membrane, and it contains a variety of cells. And actually, it contains what are known as hair cells. All right, like I said, we all have hairs in our ear, and the hairs go back a lot deeper, right? And each type of cell, okay, inner or outer hair cells, does a specific role when it comes to hearing. Inner hair cells, they are primarily there for having hearing receptors in a sense. All right, so the receptors that are there to identify the vibration and turn it into a hearing signal. All right. Outer hair cells, they are there to amplify whatever sound sensitivity is needed. Okay, so yes, the middle ear did its job, it's amplified the sounds, but the outer hair cells can be there to amplify the sensitivity as needed as well. Okay, and lastly, they have what's known as the tectorial membrane. That is more so there to help with the physiological relevance of connecting to its central nervous system. When we get to the physiology of it, you'll see what I mean. Okay, this is just a quick, again, depiction of it. You guys can take it on your own time to kind of understand the location. The cochlea is exactly what you've probably seen in your textbooks. It almost looks like a snail shell, right? So that's there for your hearing. It's got the duct to assist with as well the pressure changes and all that. As well as a variety of other canals, and that are there to basically receive the hearing, all right, the signal, the vibration, and then correlate it to the respective central nervous system, or the cranial nerve to the central nervous system. Here's just another picture to kind of put it into an idea of what exactly is occurring when you are hearing. Right, you have your ear, external, middle, internal. This is obviously the internal. Sound is coming in, hits the eardrum, vibration then occurs. So as the vibration occurs, it passes into the middle ear. Middle ear leads to an increase in sensitivity. Okay, and along the increase of the sensitivity, it starts to lead into what is known as the cochlea, right? So that's your snail shell structure. That the vibration of the sound starts to go around, go around, go around this snail shape, and this transmission of sound waves occurs from two different locations, okay, via the helicteromia. And what occurs then, as this is going forth, is this hearing receptors, they start to get fired, right? So they start to get this threshold that's needed to send a message. And now what's interesting here with hearing is that it's not just a structural location with one nerve going straight to it. It has several. They're known as amplifier nerves. Okay, so specifically when we go to our first firing neuron after sound is obtained, it's going to the vestibulocular nerve. Okay, and these I guess you can say finger-like structures that you see here, ampular nerves, are there to help with spreading out throughout the inner ear to help with sensitivity in the hearing, as well as being able to discriminate the sounds as well. All right, so looking then into further in, I guess you can say, into a little snicket section into the cochlea, we just see exactly what we mean. We have our vestibular nerve. It comes on in. This is more of the area from with the hair cells would interact. Okay, because remember we said one hair cell, its outer helps to receive the sound, whereas the inner helps to get the vibration increase, right? Or sorry, I flip-flopped those. So if I'm looking into this section on where the actual connection is occurring, this is just a mirrored image. We see that in this section of the cochlea, okay, we have our branch of the vestibular nerve coming on up, and we have our outer hair cells and our inner hair cells. Okay, so of course, like everything, there's supporting cells. We didn't go into every single cell that's in this. similar to the other ones. They all have similar rules, like the basal, epithelial, things like that. But in this instance, we're focusing on two key cells, the outer and inner hair cell. Okay. Now, if you remember, the outer hair cells, they're there to help intensify the sensitivity of the ear, of the vibrations. Okay, so they help to do their job. Okay, so they help to intensify it. And then in the process, they interact with the tectorial membrane, which the inner hair cells are attached to. So it's almost in a sense of absorption of the vibration, allowing for changes in the threshold to occur. Action potential then occurs actually within the inner hair cell, and it's that inner hair cell that actually sends the signal through that first neuron, okay, that first sensory neuron. So just like vision, we talked a little bit about development of the eyes. We're going to talk a little bit about development of the ears. These guys come around the same time eyes do. Okay, so after fertilization, about 22 days, that is when it begins. Okay, so it's not just like, now all of a sudden we have an ear. It's a slow process, and it starts with the internal ear. All right, so that's the first one to get developed, then middle, then external. All right, so in to out. And it's all contributed by a variety of different tissue types: surface ectoderm, endoderm, mesenchyme, each which helps with the development of the cavities, the canals, that eventually will become our ears. And as you see, two of those three also helped contribute to the vision or the eye development as well. Now, the internal ear, that actually begins kind of as just this placoid. All right, so it's kind of this like thick cavity. It's thick within surface ectoderms. Okay, and it was, if you're looking at a fetus, it's kind of near the hindbrain section. Okay, and now what occurs is those, it's very actually similar to kind of what happens in the eye, is those sections, plactoids, they begin to invaginate. Okay, so they form octic pits, kind of similar to what we had when we had the eye pits starting to form, right? Now these pinch off from the surface ectoderm, and then they form vesicles. All right, so we've gone from pits, and then we form to octic vesicles. And this is now officially starting to kind of build the canal system a little bit. All right, and specifically it's building it, but it's starting to embed it within a new cell type known as the mesenchyme. All right, and now those vesicles give rise to that whole cochlear duct, vestibular, semicircular ducts, every kind of labyrinth system within the cell. Okay, and whatever's on the outside, whatever that surrounding portion of the mesenchyme that wasn't so much part of the labyrinth, that gets turned into cartilage. All right, so that's, you get your cartilage in your ears, and eventually it will become more ossified, gets more of a, I guess, toughness of the internal ear. Now, that's the inner ear. Okay, so next we go off to our middle ear. Now this is a tympanic cavity, will develop first from a pouch. Okay, and it is an endoderm lined outgrowth. Okay, so specific cells, again, it's just an outgrowth of the cells, okay, of a very primitive pharynx. All right, so these auditory ossicles, they develop from the mesenchyme, as similar to what we've seen before, where they start to develop the structures that are commonly associated with the interior and or the exterior cells, or ears, sorry. Now, once the middle ear is done, they have formed this two arches. We go on to the external ear. So the external, it forms the first groove. Okay, and near you get your ears, you start having grooves, right? And an ectoderm line groove becomes the first and second arch. Additional structures within that guy, they develop from mesenchyme later on down. Okay, so what you'll start to see throughout this entire process is during the first few, all right, is centralized from one certain cell type, all right, and eventually starts to, I guess you could say, build up on itself when it comes to more complex structures within the mesenchyme, and as the mesenchyme that further then goes off to either become specialized cartilage or epithelial cells that needs to be to develop the subject. This is just a super simple idea picture of what I'm talking about. Okay, vagination kind of pinches off a little bit, and then it starts to form octic vesicles. So you start to have where your ear canals are going to start to be coming in. So this is just an idea of what the development is and how it works. Okay. But more importantly, now that you know how ears are made and all that fun jazz, is understanding how we're hearing, right? And so we have light waves, right, with vision. Now we have sound waves. And they, just like light waves, have their own kind of nature to them that determines their characteristics. Okay. So, for instance, you've all heard of frequency, right? That's all determining the pitch. All right. So, the pitch that we are determining. Now, we can hear anywhere from 20 to 20,000 hertz. Most sensitively, 500 to 5,000 hertz is what we pick up that's really sensitive. Okay. Now, intensity, whereas frequency was pitch, intensity is the loudness. All right. Hence, intensity, loud. Loudness is intense. And so that's commonly depicted as decibels. So we have a hearing threshold of 0 decibels. All right. So we decided this is what my threshold is for that I can hear. So I'm going to make that 0 decibel. 60 decibels, that's if me sitting here having a normal conversation with you. 120 decibels, that is if someone is sitting next to you and screaming into your face. Okay, so it ends up being painful. Too much of intensity for the vibration of your ears to handle or to adjust to. So instead, you actually have a different type of sensory response, and that's when your pain receptors start to come into play. So, like light, they have their own kind of characteristics when it comes to sound waves, and each changes how our hearing is registered, if it is fully hearing, if it is pain as well. It's similar to what we saw with light. Now, the physiology of hearing. Okay, so I know there was a whole crap ton of things for the ear and pictures and diagrams and fun stuff, but to put it all together and how it helps with us, well, the first thing is, as you're sitting here, you're hearing the sound waves that are coming into your external ear. Okay, and that canal brings it on down all the way into the eardrum. All right, so the eardrum acts as a drum. Sound waves hit it. It starts to vibrate the drum. Okay, and it starts to amplify the vibrations further as it gets into the internal ear, or the middle ear, the ossils, and then it gets pushed onto the vestibular window. Now, as it's doing that, pressure waves move through all of the fluids that are in the cochlear, all right, cochlear fluids, and the basilar membrane of that cochlea starts to vibrate. Okay, and as it starts to vibrate, as the pressure is starting to change, hair cells begin to bend. Now, as the hair cells bend, it starts to get the cells specifically to a point of action potential. And eventually, when the action potential occurs, that is when you get your electrical signal that is generated in the cochleal nerve fibers of that specific neuron. Now, again, picture just to look at it, right? Sound's coming in. All right, hits the eardrum. Eardrum vibrates. Okay, vibration comes all the way on up through. It hits the oculus. It goes through the 3 of them. Okay, now as it goes through and intenses the vibration all the way through, it goes into the cochlea, and in the process of the cochlea, it starts to change around the fluids that are in here, because there's 2 different types of fluids, and in the process is changing the pressure. Okay, so the fluidity and the pressure of the fluids is changing. And in the process of that changing is it also starts to vibrate the basal epithelium. Okay, so all this is occurring. Basal epithelium is vibrating, pressure is changing, and all the way on through. And what is happening is, as this is occurring, is it's leading to a vibration within the hair cells. Okay, and specifically the inner hair cells. And that's when the inner hair cells start to vibrate, and eventually we get to a point of action potential, and they will shoot off that first signal to the first sensory neurons. All right. Now, just like taste and everything else, we need to be able to, as you can say, determine differences. Okay, when we think of hearing, it's pitch. All right, and going back on what we just saw, if we're making a signal, we need to make it an electrical signal, right? We need to go from sound or vibration to electrical signaling, so we need to do some transduction. Okay, and actually they will kind of go hand in hand with one another. Now, as I said, our ability to code sound for say is all done by which region of the cochlea is getting heavy response, okay, to this process. If it's the base of the cochlea, that's associated with higher frequency sounds. If it's the apex, that's associated with a low frequency sounds. And each region of that basal membrane, which we saw is what, the sound coming through leads to a vibration of, is tonotopically tuned. Okay, so it's dependent on if it's at the base or if it's on the apex of the cochlea. Those hair cells, per se, will lead to whichever frequency sound. So that's what helps us determine the coding of pitch. Now, when all that's hitting it, and you've received the message, the sound transduction, like I had said, it happens when there's some bend in vibration into the hair cells. Okay, specifically once that gets to that point where they're impacting the hair cells, okay, we have bending of what are known as stereocilia, and it opens mechanically gated calcium channels, so it opens another cation channel. Okay, and just as we've seen before, cations come in. At this point, it's calcium, potassium, and influx then causes the depolarization. In the process of that, just as prior, voltage-gated calcium channels will open, neurotransmitters release onto the first-order auditory neurons. All right, so it's very similar to what we've seen before in the other lecture. And this whole process can change, right? The bending of the stereocilia, there can be more bending done to it. Okay, so that leads to an increase in neurotransmitter, a higher firing rate. Okay, so it can have more of a stronger response depending on how much bending is occurring. All right, and this also helps with our adaptation as well. This is just a way to put it in the picture for you. Resting hair cell. Okay, and this is touching the auditory neuron. This is just the cilia that I was talking about. If you get a signal in, something is occurring, all of a sudden the cation channel are completely open, large amount of potassium enters the cell, there's a strong depolarization, all of a sudden these calcium channels, they open up. Calcium channels open up, and what occurs is they increase more of their neurotransmitter. Calcium channels will bring it in and increase production and release. That neurotransmitter then hits that first order neuron. Okay. Now, if it's hyperpolarized, all right, so way too much polarized, it's the opposite where it's shut off. Okay, we've seen this before. This is what helps kind of with adaptation as well. All right, so this stimulation of the hair cells for hearing is very similar to what we have seen through almost every single sense. Okay. Now, auditory pathway. Okay, so what we looked at prior was more of the, I guess you can say, the ear-oriented pathway, right? The ear to first nerve. Now, what we're going to look at is known as the auditory pathway, which it's basically the big kahuna of everything. Okay, it's what happens in the ear and what happens in the central nervous system, so forth. Okay, so if I look back at the first step and, we start at the ear, this auditory pathway starts at those hair cells. Okay, so those hair cells just got hit with that transduction. All right, potassium has come in, it's depolarized, calcium channels opened. Calcium channel opening makes more neurotransmitter. Neurotransmitter leads to the activation of the first sensory neuron, or the cochlear nerve. Okay, and now the cochlear nerve goes on through into the secondary neuron, all right, the cochlear nuclei or medulla, all right, and that works its way up through superior olivary nucleus, which helps with sound localization, so localizing sound, and to the inferior colliculus, which is in the midbrain, the medial geniculate nucleus, which is the thalamus, okay, all the way into the primary auditory cortex, which is the temporal lobe. That is what helps you perceive your pitch, if you're talking too loud, the duration of the speak. Okay, but it also on the side goes to two other regions. All right, so auditory cortex is, main cone to go to, but there's two other regions it can go to and does just because of speech. Okay, auditory association area. That allows you to recognize you're talking to me, or this is music, or I know what that noise is, or to the speech comprehension area, Wernicke's area. So that's you saying, here I am, and me comprehending, you're saying, here I am, and there you are. Okay, so while it goes into the auditorial complex, it then decides, or goes in general, to both those regions. So this is just an easy, simple depiction of exactly what we had just said. Ears, nerve, up on into it, into the cochlear nuclei, into medulla, up into the midbrain, the thalamus, all the way up into primary auditory cortex. Okay, so this is just put in the picture. All right, so that was hearing. Now, hearing, just like taste, it had its play in other senses. Now, this wasn't so much a sense when I should say it wasn't so much a heavily studied intense sense when I was in school, and that is equilibrium. And more importantly, what they discovered over the years is that really hearing helps equilibrium quite heavily. Okay, and as you probably get from the name, it's just this is the ability for your body to sense and it's in a position. That sense that your body's moving, sense that it should be in an orientation, whatever space. I should be sitting against the back of the chair. That is your body's sense of that. Okay, and this is mostly done by receptors in the vestibular apparatus of the inner ear. Interestingly enough, while this is another sense, it's mostly using the ear. All right, and it's stimulated by a couple things. Three of them being this linear acceleration decretion. All right, it can change to the eutical and the saccule. Head tilt, or static position, same thing. Rotational angular acceleration or deceleration, so like spinning or turning, that consists of your semicircular ducts. All right, so those three regions correlate to each one of those three specific changes. Now, semicircular ducts only correlates to rotational acceleration, deceleration. All right, so like I said, utricle and saccule, they help with your I'm moving forward, accelerating, I'm moving back, accelerating side, side. They help with that kind of detection and understanding of the motion. All right, and so each contains a sensory region. Maculas contain hair cells, all right, similar to what we saw with ears, right? Sensory receptors, hair cells, and the supporting cells, which are basically almost the same across every single sense that we've seen so far. And those hair cells, they have the same stereocilia, okay? They have a kinocilium. That's one difference from, I guess, working with the hearing side of things. And their tip links connect to the stereocilias, okay? Now, hair bundles, okay, because it's not just like hair cells are all over within this region. They actually bundle with one another. Okay, so hair bundles are what they're referred to with several hair cells that are bundling together. These are embedded in what is an audiolithic membrane. All right, hence it gets named audiolithic organs. And they interact with a variety of things known as audioliths. Okay, and how goes. So how these audiolithic organs and the audioliths help to detect the linear acceleration, deceleration, if your head position is up, to the left, to the right, is all on certain orientations that our body takes. Okay, for the utricle, for instance, I have here, if the macula of that is horizontal, okay, that is correlating to horizontal movement in my head tilt. If it is this ensaculus, the macula is vertical, that is telling me vertical movement. So if all of a sudden that occurs while I'm on an elevator, that's my body's way of processing I'm vertically moving up right now. All right. Whereas on the utricle, horizontal movement head tilt, I'm on an escalator moving horizontally. Okay. And the mechanism is all really helped to lead by gravity or inertia. All right, so we all probably know what gravity and inertia is, but what these two come together to do is they lead to otolithic membranes to shift. All right, when I'm saying shift, I mean literally shift. And in the process of the shifting, left, right, up, down, all the fun stuff, the hairs bend. Those bundles, the hairs within them start to bend. And with the shifting of the membrane, it's going to go towards or away from the kinocilium. Now, if it goes towards it, that leads to depolarization. Now, if we go away from it, it's going to lead to a hyperpolarization of those hair cells. Now, this is the next. Remember, there's only two real senses that really play a role with hyperpolarization. This is your second one. And when it does the depolarization, hyperpolarization, flip-flop, 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 all dependent on gravity, it can change the neurotransmitter release. And so forth, since it changes that, it changes whatever signal is going to be given to the first sensory neuron, for this instance, the vestibular branch. Okay, so simply just the gravity and inertia we have are just going to change these little hairs on these cells, and that's what's going to tell your body, I'm walking forward, I'm going up. This is just a quick description, picture to kind of show you what I mean, understanding your location of where these receptors would be for the equilibrium sense. Okay, it is the utricle and the sacule. All right, so they contain maculas. This is just a more of a picture understanding. Okay, whereas as membered, these are all otholytic organelles. It's because they have this otholytic membrane. Commonly, it's associated with more calcium, but these membrane portions can correlate with a lot of hairs. So those are your hair bundles. And your hair bundles are the one that flay left, right, up, down, if they're going to directionality in which they're going to go. Down here's got a pretty good picture. As you can see, the canociliums are right up in here. Okay, and so when talking about gravity and inertia, it's not like there's much really to trick you on there. It is literally, I tilt my head straight up like this, my hair cells aren't moving, I drop my head down to the forward, the force of gravity moves that otolithic membrane, okay, which in the entails then changes the hair bundles where they're going to go. Okay, and that's what leads to hyperpolarization and depolarization. So in reality, of the equilibrium side of things, it's that otolithic membrane is really kind of the key player, because it gets taken hold of by gravity to push it to where it needs to be. Okay, so the next one is semicircular ducts. So if you remember, we had those other two, they got the up, the down, the front, the side. These guys play a role with rotation, all right? And they are just a cluster of three ducts. Okay, so semicircular ducts are not referring just to one duct, that's a cluster of three. They're all at right angles, anterior, posterior, lateral, all referring to the locations. And each one has an ampulla, so that is a structure that contains a cristae. So the cristaes are just hair cells that are embedded into more of a gelatinous structure, cupula. Okay. Now, as I said, these guys are all about rotational changes. if something's rotating, but it's accelerating or rotating, or it's decelerating. Okay, that's what these guys specialize in. And if we're talking about head rotation, for instance, it's very similar to the gravity and inertia. Your head rotates, you actually move the ducts, but you have a lag in a certain structure known as the endolymph behind. Okay, and the endolymph movement bends the cupulas and the hair bundles, similar to what we saw previously. And once again, the direction of the bending occurs. And it's all dependent on, if I'm turning left or if I'm turning right, and where, in fact, the endolymph is and where it's lagging. But it messes with those hair bundles, and those hair bundles change to either depolarization or hyperpolarization, which again messes with neurotransmitters, messes with if that first sensory neuron is going to receive a signal or not. But this we can correct, right? We can adjust. So if there's continuous rotation, eventually we get adapt and our signal stops. All right. If there's sudden stopping, all right, that's dizziness sensation, right? And it's because basically we've done it so fast that we've bended it very strikingly fast on the opposite direction, so there's not much of a bend actually occurring. Almost in a sense of you're confusing your senses to think you're going but not going. Now again, this is just a picture. Put that in here. Again, in here, you can see here, semicircular ducts. These are semicircular ducts at each tip of the semicircular duct is where there is. So again, 3 of them, right? Posterior, anterior, lateral. If you look further into those guys, this is where you'll see the hair bundles. Okay, the cupula is basically like that membrane that we had covered, or the orthotic membrane, except it's not calcium, it's channel based, all right? So it's cupula. And so we have the hair bundle, we have the hair cell, we have supporting cells. These hair cells and supporting cells, that's the cristae. You probably, when you hear that word, you probably might think of the mitochondria and all the folds the mitochondria had. It's basically, that's what this is, okay? And so what occurs is, your head's still in still position. You have this cristae, you have this bundle, you have your cupula. It's hanging up here, okay? But it's in this little alcove, you can say. But all of a sudden I rotate my head. And what happens is inertia is going to flip this little cupula to one side or to the other. Okay? And now when it's flipping left or right or whatever with the turning, it's going to bend those hair filaments. Okay? And then again, the bending of the hair filaments, depending on where it is the endolymph, that's going to determine if it's going to be hyperpolarized or depolarized. But in the process is going to tell you what neurotransmitter needs to be let off. What exactly is occurring to you? Oh, I'm turning my head to the left. I'm turning my head to the right. Okay, I turned my head too far to the right. I turned it too fast. It's just this simple gravity and inertia and changes of these small structures that are telling your body right now that you're doing that. Okay, so we basically talked about signal transduction. I have it written here so you guys, can understand it. It's all down to depolarization and hyperpolarization. Remember, this is the only sense we have that hyperpolarization plays a pretty big role. Okay, but altogether, it impacts neurotransmitter release. If it's depolarized, increases transmitter release. Okay, now if that happens, depolarized, if you remember, the sensory neurons will fire rapidly and they go through the first neuron. Okay, for this instance, for equilibrium, it's a vestibular branch. All right, now if it's hyperpolarized, this portion would not be occurring, right? Because it wouldn't increase the neurotransmitter release, so the sensory neurons would not fire. Okay, so that's why it's good that we have both of them, because we don't want constant neurotransmitter release to our body, giving us more of a vertigo issue, right? So if we look at it as the pathway, okay, now this instance, we're gonna try and kind of keep them all together, so it's not saccule versus semicircular ducts, it's all together, okay? So in the pathway, hair cells all play a key role, right? We've already pretty much established that. If they're going to hyperpolarized, depolarized. But if they'd say depolarized, we get the signal. Okay, and we get the signal. It goes through the first sensory neuron, vestibular ganglia, to the vestibular nucleus. So that's medulla and the pons. Now, this then allows for integration with a couple things: visual input and proprioceptors, especially for the ones that are neck or limbs. And actually, equilibrium is quite intense because it's not just a couple pathways going. It's actually a lot of pathways to different things. Okay, and it all goes through different tracks. 3 of the cranial nerves lead to eye movement. All right, if I know I'm turning my head to the right, then my eyes follow with me. 1 of the cranial nerves, head and neck control. Okay, I want to turn my head to the right. It's that nerve that's going, but it's also the other nerves that tell my eyes to move with it. Posture and muscle tone, vesto, poliospinal tracts. Okay, so if I'm going back to the twisting one, okay, my posture needs to stay straight, and I'm going to turn my head and my eyes with it. Okay, now thalamus, that is your vestibular cortex. That is your conscious awareness. So for me to be here, like my posture is changing, my head needs to turn, my eyes need to turn. That is all my conscious awareness that I need to do that. All right, so the equilibrium pathway is quite complex because it just integrates with so many other pathways. This is again similar to the previous, where we just gave a little explanation on what it can occur. As you can see with the blue lines, like I said, there's tons of different pathways. All right, and where it can go, and they all need to integrate and work together to determine exactly where your posture is, what the response needs to be to such changes in the environment. Each one of these nerve neurons, which we just discussed, have different roles. Now, vestibular nuclei. All right, so this integrates information from vestibular, visual, somatic receptors, all those guys. Okay, and then it sends commands to several of those regions, right? Regions that we've already kind of talked about there. First, nuclei of cranial nerves, oculomotor, trochlear, and evidence, which controls coupled movements of the eyes. Okay, so this allows the maintenance of focus on your visual field. Okay, I turn my head to the right. This tells my cranial nerves, okay, the eyes need to follow with me in a nice steady pattern so that even though I'm turning my head, I can still have somewhat clear focus of the wall that I'm looking at. All right, so it also goes to the nuclei of the accessory nerves. That's, again, your head and neck movements. Okay, so this is what helps to maintain equilibrium. All right, so while the nuclei cranial nerves is there for your focus on a visual field, the accessory nerves is there to help maintain your equilibrium, not allowing you to fall off or over. All right, and the vestibulospinal tract, that's impulses that goes down your spinal cord. Okay, so the key thing there, it is a signal or an impulse that's going down your spinal cord, but strictly maintaining muscle tone and any skeletal muscle that helps maintain your equilibrium. All right, and then lastly, ventral posterior nucleus and the thalamus. Okay, all this together. All right, so that ventral posterior, it helps you be consciously aware of your positions and the movements of your head and the limbs. Every time you walk, you turn, you're basically falling. It's just that signal you get from the hair cells and into all these nerves is allowing you to correct it so fastly that you are remaining on your feet. You're remaining balanced and you're consciously aware that I am moving because of this. My balance is here. I have turned my head this fast. Okay, so because of this nuclei, all of this is allowing you to keep standing and walking. Okay. All right, so should be the last lecture. Okay, if there's any questions, call me during my office hours, email me. I can do meeting up online. If if you haven't taken the quiz already, I hope you waited to the last one. I will be sending a password out so everybody can use that. So hopefully the lockdown browser would work. But until then, I'll see you guys next week.